Today, I am doing some practice with the micropipettes in our modern DNA lab in my department. And I have to do some practice because it has been a long time since I last did any pipetting, either in class or for just practice or for lab work. And I'm getting ready to actually start doing DNA extractions for the samples that are for my PhD dissertation. These are samples that I have drilled bone powder out of teeth that were sent to us from some archaeological sites over in Poland. So my role in this is to extract DNA from those teeth. And pipetting is one of the fundamental skills of this kind of lab work. And I really enjoy it. It's very satisfying. So I'm super happy to be back in the labs doing some practice. So the supplies I need, I have some micropipettes here. I have pipette tips in these boxes. I have a waste cup for used pipette tips, food coloring, and little tubes in which to mix said food coloring. I have red, yellow, green, and blue. And from these, I just mixed some of the red and blue food coloring, measuring it out with a pipette and then putting it into a new tube. So I have some very dark purple in here that I just did. Have instructions telling me the amount of liquid to pick up and which color, and I'm then going to mix. Next, I'm going to do some green and yellow into this. Nope, not that tube. This tube. Empty tube here. I will actually move that out of the way before I do that. If you notice, these pipette tips have little cotton filters in them, and they're the same types of pipette tips that I'm going to use in the ancient DNA lab. Those filters are in there to keep, things out that are supposed to be, not supposed to be in there. And that's a big part of the DNA extraction process, is filtering out everything from my sample that is not DNA. Because if it's not DNA, I don't want it in my sample, before I go and try to then copy and amplify that DNA so I can then analyze it later. Okay. So before I grab a pipette tip and add it to my micropipette, I've set it to the correct measurement for the amount of liquid that I want to pick up as per my instructions. I did that by twisting this knob here. Now I'm going to pick up a tip. I love doing this, with only one hand, because I thought it would be a great idea to do this one-handed so I could film. I'm going to pick up 250 microliters of the green first. So into the tube. Oh, no, that's not what I want. So I'm going to push down to the first stop on the micropipette, then insert the tip into the liquid, pull up on the knob slowly. I've picked up such a pretty shade of green though, like damn. And then move over to my other tube, push down to the second stop on the knob of my pipette there. That should get all the liquid out. Alright, well I got most of it out. And then eject the pipette tip. There we go. It would be nice if I had a buddy to help me film here, but that is not the case today. Maybe someday in the future, if there is want or demand for that, but let me know. Maybe I will recruit some people to help if they might find that fun. So one thing that I am trying to practice doing while I practice pipetting is arranging all of my supplies and equipment in a way that helps to prevent me from reaching over things, especially reaching over open tubes. Like this tube I have here that is full of my green food coloring. And that's something that we want to avoid in the lab. We don't want to contaminate anything if the tubes are open and there's stuff in there. We definitely don't want to knock things over. That's always kind of a very scary, heart-stopping moment. There's too much adrenaline, no one wants it, you feel yucky until you've calmed down. And, you know, made sure that you didn't wildly mess anything up. So that's something that I am trying to be consciously aware of so that I have a good feel for how to set up everything that I need in the Ancient DNA lab to prevent me from reaching over things, especially empty tubes. I also only have tubes open if I'm actually going to use what's in them. So if I'm not using, say, my tube of red food coloring, it's going to stay closed because I don't need it, and it's fine. So next I'm going to open up my tube of yellow food coloring and add that to the green, because that's what my practice instructions tell me to do. So here are the motions I'm actually doing up here when I'm picking up the liquid with my pipette. So I've already attached the pipette tip. I'm going to pick up my mostly greenish, green-yellow food coloring. So before I put the pipette tip into the tube, I go down to the first stop here. Then I want to go as vertically as I can into the tube, and then slowly pull up on that. There, I've picked up the liquid. To release it, we're going to go just back into the same tube. I'm going to go all the way down to the second stop. You can see it go down a bit further. And then pull it up out of the tube first, and then release that. And then this big button ejects the pipette tip into my wastebasket. And that is pipetting basics, really. You know, I find this whole process just very satisfying. I'm very glad that I've been able to come back in and practice this before I have to go do it for real in the Ancient DNA lab. But I'm hoping that'll be quite fun.